How can synthetic biology engineer new biological functions? Imagine being able to redesign living cells like building blocks to do things they never did before. That's what synthetic biology is all about. It's a way scientists can create new functions in biological systems by carefully designing and assembling genetic parts. Think of it as programming living organisms to perform specific tasks. This process starts with understanding the basic parts of biology, like genes, enzymes, and regulatory elements. Scientists treat these parts like standardized pieces of a puzzle that can be combined in different ways. By doing this, they can build new genetic circuits that control how cells behave. These circuits can make cells produce certain molecules, respond to environmental signals, or turn genes on and off. To make these changes, scientists use gene synthesis, which involves creating new DNA sequences from scratch, and genome editing tools like CRISPR. CRISPR allows precise modifications, such as inserting, deleting, or replacing genes within the genome. This way, they can program cells to do things they normally wouldn't, like producing medicines or breaking down pollutants. Designing genetic circuits is similar to creating electronic circuits. They connect genes and regulatory elements to control cellular functions dynamically. For example, a circuit might turn on a gene only when a specific chemical is present. Modular engineering is also important. It means designing biological modules that can be combined predictably, making complex systems easier to build and troubleshoot. To predict how these systems will behave, scientists use computational models. These simulations help reduce trial and error in the lab. They can test how a circuit might work before actually building it. Once a system is designed, directed evolution can be used to improve it. This involves creating many variants and selecting the best performing ones. In medical applications, synthetic biology can engineer cells to target diseases, deliver drugs, or produce therapeutic proteins. For example, immune cells can be programmed to recognize and attack cancer cells. Tissue engineering uses synthetic biology to program stem cells to grow into specific tissues or organs. Biosensors are another example, where cells detect toxins or disease markers and produce a measurable response. Microbes can also be engineered to produce complex drugs more efficiently, reducing costs and increasing sustainability. Synthetic biology moves beyond traditional genetic engineering by focusing on designing entire systems that behave predictably. It uses standard parts, computer modeling, and a systematic approach to create living systems with new functions. So, in essence, synthetic biology engineers new biological functions by assembling genetic components into systems that can perform tasks outside of natural capabilities. This approach opens up endless possibilities in medicine, research, and industry. It's like giving living organisms a new set of tools to help solve real-world problems.